In Lab 6, you are going to be looking at the process of diffusion and osmosis. So our very first experiment is about diffusion, which is just the movement of a solute from a high concentration to a low concentration until that concentration is equal, basically until we reach equilibrium. So for this experiment, you're going to be using a dialysis tubing bag. So dialysis tubing is obviously not alive, right? This is basically a piece of plastic with a bunch of little holes in it, or what we could just call little pores. So stuff can move through this bag if it is small enough to do so. Now, as opposed to a living thing like our cell membrane is going to be a little more selectively permeable because it is going to differentiate not just on size, but also on polarity and charges and stuff like that. So this is just a mimic of what we're going to do. So this tube is open on each side. So what you are going to have to do to use this is to knot it up. So the best balloon tire in your group gets this job. So what you're going to do for your first experiment, 6.1, is you're going to create a bag. This bag is going to contain starch. So you can kind of see this bag is a little bit foggy. All right, so we're going to take a water solution with starch in it and put that into the bag and tie off each end. So starch is a polysaccharide, right? So it's a large, complex molecule, and in general, it is too big to make it through the pores of this bag. So that's really what we're going to see. Can starch move through this bag? So if you will recall, starch is mixed with iodine, or iodine is used as an indicator for starch, and when it does, it turns a purplish color. So we're going to do that. So we're going to take a beaker filled with plain old boring water, all right? We're going to put the bag in here, and then we are going to dump in a few mils of iodine, right? So the iodine goes in, and it is going to sit. And as it sits, what you will see is, voila, the magic of science. You will see that the bag itself is darker than our original bag, right? So this just started, right? We just looked at it. This one's been sitting for about 20 minutes. So you can see that the bag has turned a bluish purple color. So again, remember, iodine is a starch indicator. So when iodine mixes with starch, it turns that bluish purple color, which you can see pretty well in this bag. But you will also notice that the fluid on the outside is still that color of the iodine, right? The watered down iodine. It has not changed color at all, which indicates that starch is big and cannot move through the bag, but iodine is smaller and was able to move through that bag. So right now the iodine is moving through the bag into the solution in the bag from a high concentration of iodine to a lower concentration of iodine inside the bag, but the starch can't move at all, right? So in this case, starch cannot diffuse from the bag out, but iodine can diffuse from the solution into the bag.